Welcome to today's webcast, where we will delve into the fascinating world of biospun scaffolds and their diverse applications in the in vitro research tools, or IVRT, field. I'm your host, Cameron Fainoff, and I'm excited to explore the cutting-edge technology behind these innovative scaffolds for BioSurface's IVRT business. I would like to welcome my guests for today's webcast, Matthew Fainoff, President and CTO of BioSurfaces, and Dr. Patrick Hayden, Vice President of Innovative Research. Matt and Pat will discuss in depth how our biospun scaffolds can drastically impact the way research is currently done. Before we dive into the various applications, let's take a moment to understand what biospun scaffolds are. Matt, can you provide our viewers some background about the biospun scaffolds?
Sure. Thanks, Cam, for having us. Biospun scaffolds are three-dimensional materials that are made from biocompatible, synthetic, or man-made materials. The resulting structure of the materials are designed to mimic the scaffold the body uses to grow these cells, known as extracellular matrix. They provide a supportive framework for cell growth and development, making them an essential tool in the field of in vitro research. A simple way to think about it is to think that most buildings are made of a frame that has many floors onto it, which the main structure is built. The same principle is how cells grow. They prefer a structure to build tissue, and this is served by using our biospun scaffold.
How are these scaffolds created, and what benefits do they give to researchers?
Biospun scaffolds are created using a proprietary electrospinning process. Over the past 20 years, we have developed patented processes, trade secrets, and know-how that allows the company to consistently reproduce these scaffolds. For those in the audience not familiar with electrospinning, it involves generating an electric field to the polymer solution, exciting the polymer, resulting in the polymer traveling to the grounded surface. This process results in creating ultrafine fibers that are then deposited onto the collection surface to form a scaffold. The resulting scaffold possesses a high surface area to volume ratio, porosity, and mechanical properties that closely resemble natural tissues.
What was your original vision for this scaffold?
We were, and to some extent still are, planning to use this technology for medical devices. Our biospun scaffold, due to its structure, encourages the body to heal without scar formation, also known as fibrosis. These properties, along with the ability to make different shapes and deliver target drugs, make these materials ideal candidates for use in medical devices. We conducted several different preclinical studies that have improved healing characteristics. Recently, there has been a push to make tissue outside of the body that can be used to evaluate drugs or active agents for toxicity, penetration, and in other areas. This was further brought into view when the National Institutes of Health, or NIH, requested our help for making these scaffolds for benchtop applications. This was the start of our in vitro research tools, or as you mentioned, IVRT program.
How did the initial work lead you to meet Pat and to really push the IVRT program forward?
The NIH was looking to further expand the use of our biospun scaffold for their applications. In 2018, I learned that Pat, who was working for our neighbor at the time, was an expert in making tissue models. I reached out to see if there was a way that we could collaborate using our scaffold and their plastic housing that they used for their tissue models. We began to initially examine if our biospun scaffold could grow tissue at the benchtop. In late 2019, Pat became a free agent for those sports fans in the audience. We immediately brought Pat onto the team and began to explore the possibility of providing our customers with an open source process, or more simply put, a recipe for making various tissue models. The goal is to allow the customer the ability to change different aspects of a model, like adding immune cells, active agents, neurons, and other types of changes that may be specific for the customer. The ability to provide the customer this option, along with our expertise in nanomaterials and cell and tissue biology, gives the company a competitive edge.
Fascinating. Now let's dig further into the various applications of biospun scaffolds for IVRT applications. As you mentioned, biospun scaffolds play a vital role in this process by providing a supportive structure for seeded cells to grow and differentiate. These scaffolds can be custom tailored to make specific tissue types such as skin, lung, intestine, and airway, and could even be potentially used for cartilage and bone. By mimicking the natural extracellular environment, biospun scaffolds facilitate cell attachment, migration, and nutrient diffusion, ultimately promoting tissue regeneration. Are there specific changes that can be made to the scaffold to make one type of material perform better than another? Also, what can biospun scaffolds provide that other current membranes can't provide?
Good questions. Current polymer selection is one way that we can control how different tissues grow. For example, arteries or heart muscle typically have elasticity. We can choose to grow these tissues using a polymer that stretches, such as a polyurethane. If we want the tissue to have minimal polymer remaining, we can choose a biodegradable polymer, or even further, choose a polymer where part of it goes away quickly and the other part remains to provide some type of structure. We can also alter the electrospinning process to create more or less pores in the scaffold, as well as create different fiber sizes onto which the cells will attach. By incorporating pharmaceutical agents within the scaffold matrix, and this is an area that we're just beginning to explore for our IVRT business, researchers can incorporate different biologic properties right at the area where the tissue is growing. For example, targeted growth factors can be added to enhance cell growth into the scaffold. Or alternatively, an agent such as bioglass can be electrospun into a polymer to create a bone model.
Thanks, Matt, for the biospun scaffold background. We'll now shift gears and get more specific about the end use of these scaffolds, mainly forming full-thickness tissue models. Pat, you've had a long history of developing various tissue models. Can you give our viewers a little bit of your previous work?
Thanks, Cam. My background is in vitro toxicology and tissue engineering. My early work as a postdoc at NIEHS focused on the effects of free radicals in skin cells. I also worked briefly for the Gillette company, where I first encountered organotypic skin models and used them for testing safety and efficacy of consumer products. From there, I joined MatTech Corporation, where I was a senior scientist and then vice president of scientific affairs over the course of more than 20 years. At MatTech, I led the development and commercial production of a number of their current commercial products, including Epiderm FT, Epi-Airway, including human and rat versions, as well as Epi-Airway FT and Epi-Alveolar. For the past 4 years, I have been working with Matt and his team at Biosurfaces to adapt similar organotypic models to biospun scaffolds.
What were the limitations of these previous models?
Most current organotypic epithelial models are produced on thin microporous membranes that are essentially plastic films that contain track-etched pores. These membranes are typically composed of polyester or polycarbonate, which are excessively stiff and rigid materials compared to natural in vivo matrix that cells are accustomed to in the body. This can cause them to behave non-physiologically. In addition, the film-based membranes present an unnatural barrier to passage of cells, nanoparticles, and chemicals, which is problematic for many experiments that researchers would like to perform. With regard to full-thickness tissue models, these are most often currently produced using collagen gels that are obtained from animal sources. In addition, these gels tend to be very unstable and are susceptible to unwanted contraction issues, which can be very problematic and lead to poor reproducibility. Biospun scaffolds eliminate these problems.
When you began to work with Matt in 2018, what initially drew you to the biospun scaffold?
Well, as soon as I first saw the biospun scaffolds, I immediately wondered if they could be used to create the dermal or stromal components for full-thickness skin and other epithelial tissues. The first thing we tried was with skin, and it was one of those rare times when we had immediate success from the very first try.
Now that you've had a chance to dive more deeply into working with the biospun scaffolds, what have you been able to achieve using these scaffolds in your studies?
We have also shown that the scaffolds can be used to produce full-thickness airway and intestinal models, and we have continued to refine and optimize the skin models. In addition, we were recently awarded a Phase 2 SBIR from NIEHS that will allow us to further develop the skin and lung models and validate them for specific applications, including skin phototoxicity, irritation, inhalation toxicity, and tobacco research. We will be performing this work in collaboration with the Institute for In Vitro Sciences,
Most of your work is focused on static models, meaning there is no fluid that moves. How do you envision the scaffolds being used in other benchtop studies that would use flow?
Well, another significant application of biospin scaffolds is in the field of cell culture and organ-on-a-chip systems. By culturing cells on biospin scaffolds, researchers can create microenvironments that closely resemble human tissues. These cell culture systems provide a more accurate representation of in vivo conditions with regard to the stiffness and diameter of the biospin fibers, allowing for better disease modeling, drug screening, and toxicological studies. Moreover, by integrating biospin scaffolds with microfluidic technologies, scientists can develop organ-on-a-chip systems that replicate the function of human organs, enabling advanced drug testing and personalized medicine approaches.
It's truly remarkable how biospin scaffolds are shaping the future for in vitro research. Unfortunately, that's all the time we have for today's webcast. I want to extend a heartfelt thank you to Pat Hayden and Matt Faina for sharing their valuable insights, and thank you to our viewers for tuning in. Remember to stay curious and keep exploring the wonders of science. Until next time, goodbye.